Great, so we're going to switch back to microbes for this talk, keeping with the theme of lots of different ways to make proteins. First, a little bit about Primrose Bio. We were founded in 2023. It was a merger of the Pfenex team with Primordial Genetics, a synthetic biology company. But the Pfenex Expression Technology platform's been around for about 25 years. So well established, and we have a nice facility here in San Diego, if anybody wants to come visit, where we do end-to-end strain engineering through process and analytical development to support and enable GMP manufacturing. So what we've been really focused on since starting to build this platform about 25 years ago is a way to really accelerate protein expression and identifying an optimal expression strategy for any given protein. So what we do is really combine high yield microbial expression with data-driven development cycles and try and keep that very rapid moving through to identify our optimal production strategies. And as mentioned a little bit earlier, we do have approved biologics. We've taken things all the way through the clinic and into approval with this platform. So a little bit about the platform. It was designed for protein expression. So it wasn't a lab organism that someone decided to make proteins in. We isolated this organism because it was really good at making protein. And then we built a toolbox around that. And I'll describe in a few slides how we built the toolbox and a little bit more about the combinatorial strain engineering that we do. The one really nice thing about Pseudomonas fluorescens, and the reason why we picked it, is because it is capable of high cell density fermentation. It's an obligate aerobe, and it has a lot of really nice features that make it very much amenable to large-scale fermentation. So our toolbox, there are really two parts to it. One is expression strategies, where we take two base plasmids, they're compatible with each other. Like I said, we were focused on protein production from the start and doing that for therapeutics, so no antibiotics involved in the process. These are all maintained by complementation. We have a number of different promoters, different ribosome binding sites, so we can tune transcription, translation. And then two tools that we found are really, really helpful for protein expression in Pseudomonas. One, a really large collection of different secretion signal peptides. These direct protein to the periplasmic space and can have a really big influence, not only on getting it to the periplasm, but on the titer and the quality of the protein that you produce. And then we have a growing collection of fusion partners that we use for the expression of peptides, as well as toxic proteins to help improve expression in the cell. And then we pair that with an array of different engineered host strains. So we have a pretty large bank of ready-made Pseudomonas fluorescens strains that either overexpress different folding helpers. They have protease knockouts or knockdowns, both single and stacked. And then a number of strains that we've got off the shelf, and we can build more, where we combine these folding helpers and protease knockouts together. And then more recently, we've been focused on more advanced systems for protein expression. One is non-canonical amino acid incorporation, which Pseudomonas is very good at. New tools for further improving folding of more complex proteins, disulfide bonded proteins, and then talk a little bit more in a couple of slides on efforts towards streamlining our recovery. So just wanted to show this one slide to really illustrate the power of screening a toolbox, where here we've got a subset of our secretion leaders directing proteins to the periplasmic space. We're looking at 39 different secretion leaders, as well as just cytoplasmic expression, in this case just in two different host strains. And you can see really quickly that the secretion leaders can have a huge impact not only on the amounts, but on the quality of the protein. Different secretion leaders have different levels of leader cleavage. So it's something that we want to make sure that we're paying attention to as we are screening our toolbox. In this case, we moved forward this one secretion leader number 7, where we were able to, in our 96-well expression format, get about 2 grams per liter of the protein with full cleavage of the secretion leader. Now, how we run our expression screening, since we have so many different tools we can screen, it was really important to have high throughput expression developed. So we've optimized our media, which is kind of a scale down media from our fermentation growth media, as well as how we shake, the plates we use, things like that, to optimize high cell density growth in 96-well plates. In our Pfast feasibility workflow, we are evaluating a number of our different expression tools, so we can screen about 40 to a couple hundred different vectors to evaluate different pools that can influence protein expression. We do a pooled cloning approach, so we can put all of these into our ready-made vectors, take our gene that's been optimized for expression in Pseudomonas, electroporate that into 1 or 2 different host strains, and then pick those colonies. We can use NGS to just QC our pools. After the electroporation, we select on minimal media, pick those colonies into our liquid media, and then grow in a half-mil culture. Now, in that half-mil culture, we can get to densities of about 30 to 50 ODs. So even in this little scale-down model, we're getting a lot of cell density. We can do several different assays just with a half-mil culture. In this format, we grow for about 24 hours, and then we have a standard expression also about 24 hours. Then we can lyse the cells, and then rank titer by CGE, just to look at soluble versus insoluble titer, do ELISA, biolayer interferometry, whatever assay is appropriate, identify hits. And then in this format, we're typically his tagging, or if it's an antibody derivative, it may be protein A or protein L affinity, so that we can do a high throughput affinity enrichment and enrich protein that can then be used in downstream assays. So at this stage, after about 10 days, we have purified material that we can evaluate further. Once we have those hits, we can go back, use NGS to sequence that, identify what were the tools that were good at making that particular protein. This not only gives us potentially a lead to a manufacturing strain for that particular protein, but if we're evaluating a class of proteins like Fabs or other antibody derivatives or a particular enzyme and looking at different variants, we've now identified a tool set that's going to be good for screening libraries. So we can use this workflow in a couple different formats. Now, I mentioned one of the things that we're working on is really streamlining our recovery, and the thing that would make that most effective is to get the protein out of the cell without having to lyse it. And so we've been focused a lot on extracellular localization so that we can get high quality target protein. It's really important for us to put that through the periplasm first. Most of the proteins that we're making for our partners are disulfide bonded, and disulfide bonds form in the periplasm. So we want to make sure that we get those into the periplasm. Some of these proteins are slower folding. We want to make sure that it's got time to fold before we then allow it to move out into the extracellular space. Also, moving it out into the extracellular space, we can kind of avoid proteases or other things that get released, reduce clipping or other modifications to our protein of interest. Of course, if it's extracellular, we've got simplified recovery. We can just spin down the cells. We've got lower ACPs, no host cell DNA, lower endotoxin, and that streamlines everything. Another potential feature is real-time expression monitoring. So we can just pull samples, spin out the cells, and evaluate the titer without having to lyse everything. So wanted to share some initial data that we've developed with a number of different extracellular host strains, which we've got little shorthand here, EXC. So here we're looking at 4 different proteins of varying sizes, so about 15 kilodaltons to about 60. This is 96-well expression data. where we're looking at 3 different extracellular hosts. So these have different host cell modifications that allow for the protein to move out to the extracellular media versus our just standard P. fluorescens host strain. The cell pellet is just, we're going to spin down the cells, separate the cell-free broth or CFB from our cell pellet. The cell pellets are lysed, and we run out the soluble fraction just to see what's left inside the cells. And then look at the extracellular media, the cell-free broth as well. And you can see for these different test proteins, depending on the protein, different host cell modifications are allowing that protein to get out into the cell-free broth. So we're finding that a number of these modifications we're making are allowing proteins of varying sizes, varying types to get out into the extracellular media. Of course, it's really important that the quality of that protein is good, so we can test activity. We also looked at intact mass for each one of these, so you can see for each one of these test proteins, so 2 different antibody derivatives of different sizes, an enzyme, and modified microbial toxin. Each one has really high quality, so we're not seeing clipped products. We're seeing intact mass as expected, and really nice quality out of this 96-well format. So now I'm going to shift gears and talk through a couple of different examples of proteins that we've expressed with the Pfast workflow or standard workflows. This first case study is a Fab VHH fusion, where we use our Pfast workflow to evaluate the scaffolds and select strategies for scale up for manufacturing. And what we found is that Pfast workflow combined with the platform allowed us to identify expression strategies that give us high titers of soluble Fab VHH that are active, and now we're ready to move those forward into scale up. So how do we do that? First, of course, we want to optimize for codon usage in P. fluorescens. And for those who've worked with different antibody fragments before, identifying the best way to set up the operon for expression in microbes is really important. So we wanted to evaluate either a heavy chain first or light chain first operon strategy. On the second protein of the operon, we're going to fix a single secretion leader. And then we clone that. We use a standard Golden Gate-like strategy to clone into all of our different vectors. So you can have one gene synthesized for each strategy of operon design. Pop that into 40 different expression vectors where we're evaluating a number of different ribosome binding site, secretion leader combinations. Then with the Pfast workflow, we're just going to electroporate that into a single host strain, collect our pool, and then evaluate expression and binding activity of the Fab VHH. So after the Pfast expression, our sample processing workflow is to just harvest the culture. We typically dilute by factor of 3. We use sonication to generate our soluble lysates, and then fractionate into soluble and insoluble, and then evaluate using SDS-CGE so we can get an idea of the quantity and the quality of protein that we're making. Do ELISA to evaluate the activity, and then LCMS to ensure that we don't have any clipping. So this particular Fab VHH did not have an interchain disulfide, so we needed to look at this under just reduced conditions and evaluate the heavy chain VHH fusion, which is the top band, and then our light chain. And you can quickly see here with the different strategies that we're able to rapidly find expression strategies, particularly with the light chain first in the operon, that give us a nice balance of heavy chain and light chain. We then wanted to quant that. We were finding that we were getting about 0.5 gram per liter of Fab VHH in our half-mil cultures. So when we plot that out, we want to be able to compare the light chain and the heavy chain against our ladder and look at the balance of heavy chain and light chain that are made under each one of the strategies. And so we've got that plotted for each one of the operon designs here. And you can see the light chain first, then heavy chain VHH strategy was giving us much higher titers. So a little bit over 0.5 gram per liter. We then selected 48 different strains that were screened with these open triangles or open diamonds to evaluate by NGS to see what were the expression strategies that were giving us those high titers. And then we took a subset of those to confirm by intact mass. We then evaluated these by ELISA. So we did a single dilution, tested that on ELISA to assess the binding activity to the target antigen. And again, here confirming that the light chain first strategy is giving us much higher titers. The ELISA kind of pinged out at 360 megs per liter, just based on the standard curve that we used. But we were able to see that those strains that were giving us high yield by CGE were also giving us high yield of active Fab molecule. And then when we evaluated those by intact mass, you can see here on the right-hand side, this is our Fab VHH reference, where we get a nice single peak on mass spec. When we looked at the enriched material, the Fabs from a couple of different samples, we can see that we've got intact assembled Fab VHH, where heavy chain and light chain have assembled together, and then a little bit of free light chain, heavy chain down here in strain 2, where we had the light chain first, versus a lot of free light chain in the light chain second in the operon strategy. So again, confirming that that light chain first operon design was much better for this particular Fab. So hopefully in that example, you're able to see that with that fast workflow and the Pfenex platform, we're able to really quickly run through a number of different expression strategies that'll give us, very high yield of Fab molecule very quickly, so in less than 10 days. This diverse screen allowed us to identify expression strategies that work well for this particular Fab. We've worked with other Fabs where heavy chain first is better, so one operon design does not always fit for every single Fab molecule. But this screen allows us to really quickly get to a clear path to give us multi-gram levels of enriched Fab. When we scaled this up to our 2-liter scale, all of that was completed in about 5 weeks. All right, so now I'm going to switch gears. Still an antibody derivative, but this is more of a peptide, a complex peptide, where we used our platform to find tools that were going to be useful for expression of a very small disulfide bonded peptide called a picobody. We screened a number of different fusion partners and also non-fusion approaches to expression strategies, and were able to successfully scale, purify active peptide at about 1 to 4 grams per liter at the 2-liter scale. So first, a little bit about picobodies. These are really small portions of cow antibodies. So it is the knob portion of the CDR3 on the heavy chain. And it forms sort of like a little knotted structure. These are about 4.5 kD, so very small, and they can contain multiple disulfides, so typically about 2 to 4 disulfides. And the nice thing about these knob portions is this portion alone will impart binding for the molecule. So it can bind into little cryptic epitopes, into small pockets in proteins. So really unique little molecule for using for therapeutics or diagnostics. So we were screening a number of these different pico bodies. So we looked at about 4 of these. Again, 2 to 4 disulfides. These are typically about 4 to 6 kilodaltons. And we evaluated a number of different expression strategies. So either with and without secretion signal on fusion partners or without fusion partners, and then evaluated a couple of different engineered host strains. Our workflow is very similar to what I already described for the Pfast workflow. So we did our expression screen. We then evaluated binding, scaled that up to look at robustness of the selected expression strains in our 2-liter fermentations, and then did enrichment and cleavage if it was necessary. So in the screening, we looked at different fusion partners. These are Pseudomonas-derived fusion partners. They have linking it to the pico body, a cleavable linker with an affinity tag to just enable enrichment. These can boost expression, improve solubility. It allows us to infinity enrich, so very handy for small peptides. Because these pico bodies and their structure are so stable, we also wanted to look at, could we express this thing all by itself? Typically, when you express peptides in an organism, they get chewed up, but we wanted to see whether, because of the innate properties of the pico body, we could express these on their own and be able to enrich them from Pseudomonas. So at first, we looked at the pico body with an affinity tag, and then I'll show you some data later where we looked at it without a tag. So here is the output of our 96-well screen on the left-hand side. So we looked at about 800 different expression strategies for the 4 different pico bodies. We're looking at results for 2 of the different pico bodies here, looking at 2 different fusion partners or his tag only, so no fusion partner. And you can see here, this is the antigen binding activity. So we use BLI to just screen through and identify which strains are giving us binding activity, expressing properly folded pico body. And the first thing you notice is that with the fusion partners, we're actually seeing nice activity. So the fusion partner itself is not impeding the activity of the pico body, so that's nice. And the other nice thing is that even if we only have a his tag on it, we're able to detect nice binding activity from these strains. So then we took a subset of those strains and scaled those up into our 2-liter fermentations. This is a high cell density fermentation. We looked at a couple of different conditions, and you can see here in the orange bar, we're looking at our whole cell broth. So basically, are we expressing protein in the soluble fraction versus is anything getting out of the cells, so in the cell-free broth? And you can see for a number of these strains, we're getting really nice binding activity. So this is just relative binding rates. So nice expression, whether it has a fusion partner or is just a his tag protein. So we're getting a really nice amount of protein, small peptides, even without using a fusion partner. We then wanted to look at some of these non-fusion pico bodies, just scaling them up to evaluate, do we need a tag or no tag? And can we optimize this fusion-free version of pico body expression? So on the right-hand side, again, looking at BLI here now, since we're able to enrich and have a standard, we've got our titer, and you can see that for certain pico bodies, we're able to, under our higher temperature conditions, see really nice over 1 gram per liter titers of just the pico body itself. We are seeing some of that protein move out into the cell-free broth as well. We also found that for some of the proteins, in particular, for expression in this strain 1, R2G3, the high temperature combined with lower pH was more optimal. And again, here just plotted out so you can see the time course. We get really nice induction of the picobody with the higher temperature induction. So then we moved our good fermentation condition, expression strategy cultures into our purification. Here, looking at both getting that out of the cell culture, the whole broth, versus using what's in the cell-free broth. Cell-free broth gives us a nice way to remove a step from purification. But you can see on this left-hand side, this is first looking at our clarified lysate. You can see we've got some of the protein expressed down there, but we're able to use ultrafiltration as a method to just, without an affinity tag, enrich the picobody and then get that onto cation exchange for further purification. And we're able to enrich the protein. Then if we look at the material from the cell-free broth, so here looking at the cell-free broth concentrated and then filtered, we can see that we're retaining a nice amount of the picobody. Then putting that over our cation exchange, we're able to purify that away from any residual host cell proteins in the cell-free broth. So here, hopefully been able to show you that there's a really nice path to not only expressing these peptides without a fusion partner, but we're able to use the extracellular localization to remove the homogenization step and develop a method that will be scalable for later manufacturing. Of course, most important, these are active. So we were able to show that they were able to bind the RBD domain and of SARS-CoV-2 and block ACE2 binding. So really important to show that not only could we express them, but they're maintaining the activity as expected. So just a couple of key points to summarize there. Really high titer expression of a pretty complex peptide using our diverse expression screen, and really quickly able to develop a clear path to manufacturing. So just a couple of notes about the platform itself. This is a very safe platform. It's a biosafety level 1 organism, Pfluorescens. It's been in the clinic a number of times. There are 6 approved products. So really nice expression system for high level, high titer expression of very high quality protein, and really allows you to accelerate development by getting through and identifying very quickly an optimal expression strategy for your protein. I can talk to folks about this later, but we've expressed a wide variety of proteins over the last 20 plus years and consistently seen about 80% success rate when proteins have failed in other systems. So really able to show higher yield, higher quality compared to many of the common expression systems out there, E. coli, CHO, et cetera. Yeah. So with that, I'll close and just say we have a booth if you guys want to visit us later this week. If you want to learn more about the Pseudomonas system, we have 2 of our scientists here presenting posters as well.
All right. Thank you very much, Diane, for your presentation. We are out of time at this point. Oh, one? Okay, go ahead.
I don't have one, so
Oh,
let's get one.
I thought you said we have, okay.
Yeah, we have time for one question.
One question.
Yeah. I have a question. I want to thank you, Diane, for bringing up recombinant peptide expression. We've added a new pipeline on that topic here. Traditionally, it's chemical synthesis. So how are you encouraging the peptide community to take a look at recombinant peptide synthesis?
Yeah. I think there are two things. One, as peptides get longer, synthesis is the cost of synthesis, getting rid of all the organic waste streams, all of that get really high. So recombinant expression, whether it's with a fusion partner or not, can offer really nice advantages, especially when you can get to super high titers and an easy recovery process. The other thing is in the incorporation of different non-natural amino acids. That is something that we are working on, both non-natural amino acids for click chemistry, for stabilizing proteins and peptides. And so that can be an advantage for us as well. That opens up the opportunity for recombinant expression, whereas people thought that they needed to do synthesis, chemical synthesis for those in the past.
Anything else? Okay.
All right, thanks.
Well, thank you very much. I'm sure you'll be around as well for additional questions here and at the booth with your colleagues. Thank you very much. We are taking a networking break right now, and we ask everybody who's going to come back to be back by 3:30 for our next talk. All right, thank you.